Howdy folks, welcome to chapter 13, characterizing and classifying viruses, viroids, and prions, all of which are acellular, meaning they're not cells. So this cartoon is SARS-CoV-2, severe acute respiratory syndrome, that's SARS, and then CO is corona, and then V is virus, and 2 is because it's the second SARS-CoV critter. It's not really a critter because it's not alive, but the SARS-CoV was initially named, and maybe they didn't think there was going to be a 2, so they just called it SARS-CoV. But there is a SARS-CoV-1 and a SARS-CoV-2. And this is a strain of coronavirus that causes that disease called COVID-19. So this blue thing, whole thing is the virus. The most superficial aspect, you have the outside of the virus, and it's covered in these attachment proteins that kind of look like blue tulips. They're called spikes. And then deep to the spikes of the virus is the single-stranded RNA, which is the type of virus coronavirus 2 is. It's a single-stranded RNA virus. And then you have down here at the bottom the host cell. That would be one of your respiratory epithelial cells. So here's the plasma membrane, so the phospholipid bilayer of one of your respiratory epithelial cells. And in that plasma membrane, or the made of the phospholipid bilayer, you have these receptors. This green one is called ACE2, and the purple one is TMPRSS2. These are sticking out. These are receptors, they're proteins sticking out of the cell membrane of your respiratory epithelial cells, for example. And you can see in the cartoon that these spikes on the virus will recognize the ACE2 receptor stuck in your cell, and when the two bind to each other or recognize each other, that's called attachment. Once that happens, this TMPRSS2, the purple thing, can activate the virus such that the virus can go from being outside of your cell to inside of your cell. So viruses, viroids, and prions are all acellular. That means they're non-cellular. That means they're not living. They all lack their own cell membrane. So they may take a cell membrane from an actual living cell that it was in, but in terms of their own phospholipid bilayer, they don't have that. Viruses are ultramicroscopic. That means you have to use a transmission electron microscope to see them. They're measured in nanometers. They require a host for replication. That means they require a host cell. For example, in the case of the coronavirus, needs the host would be one of your respiratory epithelial cells. They get inside that, then they can replicate. They can make more of themselves. This means that they are obligate intracellular pathogens. So they are obligated to use your cells to make more of themselves. Viruses in general can infect humans, bacteria, and plants. Viruses, viroids, and prions together as a group, they all lack most of the characteristics of life. That means they cannot carry out metabolic pathways. So when you saw this word metabolic, I hope you thought glycolysis, Krebs cycle, and electron transport, because we talked about that previously. Viruses cannot grow. They can make more of themselves, but they can't grow. They cannot respond to the environment. So, for example, if a bright light is shined in our eyes, we respond by, closing our eyes or turning our head away. We're responding to a stimulus in the environment. They can't do that. They cannot reproduce independently. That just means they have to use a host cell to make more of themselves. So let's look at this cartoon on the right side. So the most superficial, you have the lipid envelope here. So this could be a phospholipid bilayer, a modified phospholipid bilayer that was taken from an actual cell that the virus lived in. And then within the lipid envelope, you have these envelope proteins. In the previous slide, we called them spikes, because it's these envelope proteins that will recognize receptors on your cells. And then deep to the lipid envelope, you have this whole area called the tegument. And then deep to the tegument is full of proteins. Deep to the tegument, you have this thing called a nucleocapsid or a capsid. It's the same thing, a capsid or a nucleocapsid. Just holds the nucleic acid inside. And in this cartoon, the nucleic acid happens to be DNA. But you could be also an RNA virus. So you could be a DNA virus or an RNA virus. And then within those two categories, you could be a single-stranded or ssDNA virus, or you could be a double-stranded or dsDNA virus. Same thing for RNA. You could be a single-stranded RNA virus or a double-stranded RNA virus. So here's another cartoon of a virus. And now in this cartoon, they're calling the spikes glycoproteins. And really, a spike is just a general term. What is the spike made of? It's made of glycoproteins. What's a glycoprotein? A sugar protein. So a carbohydrate protein. So glyco is carbohydrate, protein is protein. So these spikes on the outside of the virus are made of glycoproteins. And then the spikes are pretty much embedded in the envelope. So this is a modified phospholipid bilayer called the envelope. Deep to the envelope of a virus, you have matrix proteins, these lime green things, and then deep to the matrix protein, you have the capsid. And this cartoon, they're telling you that the capsid is helical. That's its shape. And then within the capsid, you have the DNA or RNA of the virus. Technically, if a virus is outside of a host cell, so it's just living on the counter, then it's called a virion. Once the virion gets inside the host, it's called a virus. But I think in general, you just can use virus and virion interchangeably, but in this class, you should know the difference between the two. The protein coat that covers the nucleic acid is called a nucleocapsid, so we looked at that. So this is in this cartoon, it's called a helical capsid because that's the shape of the capsid. A nucleocapsid is just any capsid, regardless of its shape, that contains the nucleic acid, either RNA or DNA. Some viruses come with this lipid envelope, this phospholipid bilayer, some don't. If you have no lipid envelope and you are a virus, then you are naked. So go get an envelope. The capsid, which is this part of the cartoon right here, is primarily responsible for the shape of a virion. So remember, the virion is just a virus that's outside the cell. So again, here we're talking about the viral capsid morphology. So this blue thing is the capsid. Its shape is helical. And each one of these little blue units, that's a capsomer, and it's made of a protein. And then this is a transmission electron micrograph of an actual virus. So it doesn't have the numbers on here, but this could be as small as 1 nanometer. So viruses are very tiny. So here's our coronavirus 2 cartoon. The outermost layer of a virion, so this actually would be a virion because it's outside the host cell. The outermost layer of the virion is a protein capsid. So this blue thing right here on our coronavirus, this would be the capsid. And the capsid provides, or if you had something superficial to the capsid, that would be the lipid envelope. Okay, so in this cartoon, the capsid or the lipid envelope, whatever structure contains these spikes, would provide protection and recognition sites to bind to the host cell. So these blue tulip spikes provide recognition sites so that the spikes can bind to these receptors in the plasma membrane of the host cell. In this case, that receptor is called ACE2. So if you are a viral spike on a virus, you serve the following functions: to be able to recognize a host cell, which is in this cartoon. And once you recognize the host cell, that's called attachment. And then after that, if you're a virus, after you attach to the host cell, then you can get activated. That will allow you to enter the host cell. And then once inside the host cell, you make a whole bunch of yourself. And then you exit the host cell. And where one virus went in, maybe 100,000 viruses came out. And you start to get really sick. So there's a term called host specificity of a virus. And that just means that a virus can specifically infect a single host specificity. So, and that's due to the interactions between the viral molecules, which in this case would be the spikes, and the host cell surface molecules, which in the case of coronavirus 2 would be these receptors called ACE2 found in respiratory epithelial cells and other cells too. So it says here in this first bullet, host specificity of a virus. Most viruses infect only particular host cells. And then let's take a look at the cartoon at the top. Step 1, here's a virus. So it looks like it's just in its capsid. This virus doesn't, at least in the cartoon, doesn't look like it has an envelope. Here's this purple stuff is the nucleic acid on the inside. And then here, these kind of orange things, these are receptors on the cytoplasmic membrane of the host cell. So step 2, the receptors, or the spikes on the capsid of the virus, recognize the receptors on the cytoplasmic membrane of the host cell. And then you have entry of the virus into the host cell. So this is called direct penetration. In the second cartoon, letter B, viruses can use membrane fusion. So in this cartoon, it looks like this virus has an envelope, and there's viral glycoproteins sticking out of the envelope. Here's the receptors. These orange things are the receptors on the cytoplasmic membrane of the host. So the glycoproteins recognize the receptors on the host cell, and the envelope of the virus can fuse with the phospholipid bilayer of the cell membrane. Because remember, the envelope of the virus is a phospholipid bilayer. The membrane of the host cell is a phospholipid bilayer, so you can get fusion between those two phospholipid bilayers. And then the virus now, this purple thing, the viral nucleic acid is free to get inside the cell. Okay. And then in step 4, it says the viral glycoproteins that were on the virus remain embedded in the cytoplasmic membrane of the host cell, and the nucleic acid of the virus in the capsid, excuse me, gets inside the cell, and then the capsid uncoats or breaks down, and the viral genome now is free to get into the cell and start making its own viral proteins. So we've said this, once inside the host, the virion becomes a virus, and the envelope is left on the surface of the host cell, and then the protein capsid and the nucleic acid is inside the host cell, and the protein capsid is removed, and then the nucleic acid can be free in the cytoplasm of the host cell. So hosts of viruses, you can be a cellular host, you can be a whole tissue host, or you can be a whole organism host to viruses. So in this first cartoon, cellular tropism just means tropism just is target. Here's the virus with its spikes on the outside. In cellular tropism, the spikes on the virus recognize receptors on macrophages, but those spikes on the virus do not recognize any receptors in neurons. So, for example, human immunovirus normally infects macrophages because the spikes recognize receptors on the macrophage, but they do not. HIV normally does not infect a neuron. Tissue tropism is kind of a higher level. So, for example, the influenza virus will infect the tissues of the lung, not a specific cell of the lung, but it won't affect brain tissue. It will not affect brain tissue. And then in host tropism, there's a myxoma virus. It normally can infect all the cells of the rabbit, I think, but I'm not sure about myxoma. But in host tropism, the virus can infect a rabbit, but it will not infect a human. So HIV infects humans. So if it's stated that way, human immunodeficiency virus affects humans, that would be an example of host tropism. But if I said human immunodeficiency virus infects macrophages, that would be an example of cellular tropism. So animals, plants, bacteria, and archaea all get viral infections, and even a tiny virus infects another virus. That's amazing. If you are a virus and you infect bacteria, you are called a bacteriophage, or just a phage for short. So a bacteriophage or a phage is a virus that infects bacteria. And so here's a transmission electron micrograph. Here's a bacillus, a bacteria called a bacillus, and these little purple things, those are bacteriophages trying to inject their DNA into a bacteria. The first virus ever discovered was a plant virus called tobacco mosaic virus, and here's what it looks like when a tobacco plant leaf has tobacco mosaic virus. It looks like this, and then here's a healthy tobacco leaf. Here's a scanning electron micrograph of these blue things. This is a scanning electron micrograph that's colorized, so these blue things are the viruses, and then this kind of brown stuff, that's just the tissue that the virus is infecting. And here's a transmission electron micrograph of HIV budding off of the cell that infected. This is genetic material of viruses. So first of all, I want to just go over the central dogma of biology is that DNA, and here's a cartoon of DNA, the double helix with A, T, C, and G, makes RNA, and then RNA makes protein. So we've gone over this in previous lectures. When DNA is making RNA, that's called transcription. When RNA is making a protein, that's called translation. And then this is a transmission electron micrograph of the E. coli genome. So all this stuff, this is all circular DNA of E. coli. And then this little piece right here is the genome of a bacteriophage, aka a bacterial virus. So host cells, that's our cells, for example. Our eukaryotic cells, have double-stranded DNA for genome. So we have 23 pairs of chromosomes, and they're double-stranded in our genome, in our human genome. And we talked about this already. If you're a viral genome, you can have single-stranded or double-stranded RNA or DNA. We said the genome may be linearized, that's like our genome, or it could be a circular molecule, that's like the genome of the bacteria E. coli. It's a single circular molecule. And then it just gives you some examples. Influenza virus has 8 linear segments of single-stranded RNA. Here's a cartoon. There's a lot of work done on viruses and how to group them. So in this cartoon, I found that you can group viruses as 1, 2, 3, 4, 5, 6, or 7. And then it shows, so for example, group 1 here, it shows group 1 is a DNA virus, a group 1 DNA virus. And then you could, if you're in that group, you could be a plus strand or a minus strand. And then coronavirus is a RNA virus, and it's a plus strand RNA virus. So, and it's a single-stranded RNA virus. So in this cartoon, it shows group 6. It shows a single strand of RNA, and it's a plus strand. And it shows here that it needs reverse transcriptase. But actually, I think that the coronavirus does not need reverse transcriptase. So I think the coronavirus is a single-stranded RNA virus that's a positive strand virus. But if you are a virus and you're in any one of these groups, all roads lead to Rome. That means at some point you all, through your fancy biochemistry, make a messenger RNA, and then that messenger RNA gets translated into viral proteins, and then the viral proteins get put together and you make more viruses. And you do this all within a host cell. So you basically take the host cell machinery for making proteins and you use your DNA or RNA to make messenger RNA. That messenger RNA is viral messenger RNA that goes on to make viral proteins. So here we go. Here's the genetic material of viruses based on the different classes. And here's Coronaviridae. So that's the type of virus that the coronavirus is. So it's a class 4 single-stranded RNA virus. It's positive RNA virus. And I did a quick search on coronavirus, and I believe that it is a polycistronic messenger RNA virus. So we won't go into the details of that, but if you're interested in this, you can read further. So here's our cartoon of coronavirus. We said it's a positive sense, so it's got the plus sign in front of it, single-stranded RNA virus. And the positive sense viral RNA genome can serve as messenger RNA and be translated into a protein in the host cell quickly. So you don't need reverse transcriptase to do a bunch of biochemistry that would take time to make a whole bunch of viral proteins. Positive sense viral RNA genome goes into the host cell and it serves as messenger RNA. You can start translating that messenger RNA into viral proteins right away.